Our next presentation is Affinia Therapeutics, and I'd like to welcome Rick Modi, CEO, to the stage.
Good morning, everybody, and thanks for attending the presentation for Affinia Therapeutics. By way of quick background, I'm the CEO for the company. I'm the founding member of the team. Prior to Affinia, I was at Avexis, where I had the pleasure and the privilege of bringing Zolgensma to patients with spinal muscular atrophy. For those of you that know about the story behind that drug, it's a one-time therapy. It's given to babies who really, when they're born and have the mutation for SMN, know that they have a less than 2-year lifespan left, right? And with this one-time therapy of Zolgensma, these kids look, act, and enjoy life just as being normal. Our vision at Affinia is very simple. We want to bring that magic to many other populations in the world, not only for rare diseases, but also prevalent diseases as well. And I'll walk you through some of the progress that we've made. The way we've tackled the problem is to realize that the biggest issue in gene therapy to go from small, ultra-rare diseases to larger, more prevalent diseases is one of targeted delivery. And the two tissues in which this is most limiting are the CNS and muscle. And in both these two areas, we made tremendous progress over the last 2 or 3 years. In the heart, we are now able to get to north of 90% of cardiomyocytes. This is NHP data that I will walk you through. If you compare that with what conventional capsids like AAV9 can do, it's less than 50%. And the doses that we use are very modest. In the brain, if you look at what conventional capsids can do, it's less than 5% of neurons. That's not enough to make a dramatic difference in gene therapy in many diseases. With our new capsids that we have, we're able to get to north of 50% of neurons. And behind this is a platform that we call ART, or Affinia Rationally Designed Therapies, where we use machine learning, barcoding of capsids, screening methods, NGS, and then a structure-activity relationship-based method of selection of these capsids. It's exactly what people have done in biologics and in small molecules over the last many decades, and we're taking that same paradigm and bringing it to AAV capsid development. We do have a very nice deal that we've done with Vertex Pharmaceuticals for Duchenne muscular dystrophy and myotonic dystrophy, where we delivered to them the capsids that they had predefined criteria for us and won the maximum financial milestones. So let's start first with the cardiomyopathies and what we can do in the heart. The diseases that are listed on this slide are examples of diseases that are genetic in their biology. Tens of thousands of patients in the US that are affected. And in addition, these patients are also affected in the prime of their lives. More importantly, for a small biotech company like ours, and many of you, when you do a first-in-human trial in these diseases, the nice thing is that you can biopsy the heart, you can show your transgene being expressed and the protein that is defective being produced, and you can see effect size, at least signals of effect, within about 6 to 12 months of the trial in a very small number of patients. And if you look at the data, as I will show you, for what AAV9 and other conventional capsids can do, you can get to enough number of cardiocytes if you give the dose at something like 1E14 vector genomes per kilogram or higher. But that dose, unfortunately, in humans is not tolerated. So let me walk you through some data that is on this slide, which is a non-human primate study. There are 2 capsids that are Affinia capsids that I'm showing you, 108 and 109. 108 is liver detargeted, and there's a legend on the very top right that shows you that, and 109 is not. If you forget to look at the legend, you can just look at the liver slides over there and see the remarkable difference, by the way, as well. This is immunohistochemistry slides reading out GFP. This is the study that we did in NHPs that was a head-to-head comparison. If you start with the 6x6 panel on the left, and you start with the lowest row, which is for AAV9, the conventional capsid, you can see that at this dose, the 1E14 dose, which isn't really tolerated in the clinic, in NHPs, AAV9 actually does get to the heart. It also gets to the liver and the DRG, which is not a good thing. If you look at our capsid 109 that is over there, you can see that we get to the heart. In this case, because this one is not liver detargeted, we also get to the liver because the backbone here is an AAV9 backbone with a cardiotropic peptide put on that backbone on the VR8 region. But you can see that for this capsid, we already detarget the DRG. And then lastly, if you look at the capsid, that is the Affinia capsid 108 on the top, this is one where we had the cardiotropic peptide put on the AAV9 backbone and a second mutation on the VR1 region where we detargeted for the liver. And you can see remarkably how much we detarget the liver and the DRG with both these. You might think that is actually very, very exciting data. The exciting data is actually on the slides on the right-hand side. So this is the 2x3 image on the right that you see at a lower dose. So this is the head-to-head comparison for these three same capsids, but at a dose that is actually tolerated for AAV9 in the clinic, 1E13 vector genomes per kilogram. And you can see, again, if you start with AAV9 in the heart, at that lower dose, it just drops off a cliff, which shows you why AAV9 in the clinic is very difficult for it to show the effect size in many different cardiotropic diseases, in many different genetic cardiomyopathies. And then if you look at 108 and 109, the two Affinia capsids, you can see that at that lower dose of 1E13 vector genomes per kilogram, we remarkably are able to get to the majority of the heart cells. This is the same data, but quantified. So on the left, we show you the lower dose that is tolerated in the clinic, 1E13. You can see 108 and 109 get to the majority of cardiomyocytes. The AAV9 bar is there, but it is a very small bar, showing you that it is insufficient for the number of cardiomyocytes that need to be transduced. On the other hand, for the chart that is on the right, you can see that at the higher dose that is not tolerated in the clinic, AAV9 could actually get to the bulk of the heart cells if that dose had been tolerated. This is the same study, but now showing you from the percent of cells that were transduced in the last slide to this one where you see expression. So in this case, we're showing you RNA. Mind you that the y-axis here is a log scale. You can see that 108 and 109, the two Affinia novel capsids, are log magnitude better than AAV9 at both doses that are shown here. We are, of course, planning on taking this capsid and moving them forward into development therapies. For those of you that are attending ESGCT in a couple of weeks, we'll show you two genetic cardiomyopathy models, and I think also a heart failure model as well, where in both these cases we have great effect, we have safety, and we have significant differentiation from AAV9 in all these genetic cardiomyopathy models that we've tested so far. The same story in CNS. So in this case, again, we're looking for diseases that affect tens of thousands of patients in the prime of their life. Unlike the heart, of course, you cannot biopsy the CNS, but you can look at biomarkers that are quite well accepted by the agency, such as neurofilament. And on the very right, you can see the AAV9 capsid is insufficient to get to the brain. For the case of Zolgensma, when I was at Avexis, we were able to get there in kids because these kids are newborns. Right after birth, the blood-brain barrier is highly permeable. It matures very quickly, and it loses that permeability. So in young adults, in young kids and young adults and adults, AAV9 is insufficient to get to these patients when it's given IV, which is why many companies try to use a local delivery. But again, when you use a local delivery, whether it's intraparenchymal, intrathalamic, lumbar puncture, intracisternaal, in all of these cases, you can get the proximal cells being transduced, but they incur toxicity, unfortunately. And the distal cells, you're back to square one, meaning you have less than 1% of the neurons further away from the injection site being transduced, which is not good enough. What we do here is very similar to what we do on the heart. We start with an AAV9 backbone. On that backbone, we put a peptide that is tissue targeting. In this case, obviously, we rationally design and screen for peptides that are permeable through the adult blood-brain barrier. And where necessary, we can put in a second mutation to liver or DRGD target or to get a second attribute that we would desire. This study is, again, in non-human primates. In this example, we did not use AAV9 as a comparator because we've done multiple NHP studies with AAV9, and all of them are less than 1%. You can see the dotted bar here, which is what AAV9 has shown from previous studies that we've done, and which is very widely accepted in the industry. In the case of our capsid, in this case, it's called ATC-134. No matter which region of the CNS, spinal cord, even including the deep brain regions that you look at, we get to north of 50% of neurons, which is very remarkable. Same thing, we want the cells to be transduced, but we also want them to be expressing. In this case, you can look at the RNA expression. Here, what we did is a microRNA approach. So what we're looking at here is the expression of the MER in these different regions. And again, compared with AAV9, we are at log order improvement. By just putting the neuropeptide on the AAV9 backbone without even putting a second mutation on the AAV9 backbone, we were able to detarget both liver and DRG in this case. So unlike muscle, where we showed you the slide with AAV9, where you see more than 90% of those cells being transduced with the brown staining in that case for GFP, in this case, you can see with ATC134, you don't see any staining at all. It's less than 5% of the hepatocytes or the DRG cells that are transduced with ATC134, providing not only the effect size that we would desire for a gene therapy, but also a safety advantage as well. You might wonder how we're doing all this. As I mentioned, the platform is called ART, or Affinia Rationally Designed Therapy. What we do is bring classical drug discovery to AAV. We make structural changes on the AAV, whether it's the peptide or the backbone. We conduct experiments to look at how that structure change makes a change in the attribute of that capsid. That attribute can be tropism, manufacturability, immunophenotype, or translatability. Just to give you an example, when we look at tropism, it could be an NHP tissue that we harvest and look at, or that we detarget that we want to look at. It can be a humanized knock-in mouse model, or it can be a bead-based assay for receptor binding. It can be any of those things. It's that versatile. And the same types of attributes can be measured through varying different number of methods for all the other three attributes that I've listed there, including manufacturability as well. We are now able to get manufacturing, by the way, even for AAV9, to the level of cost of goods sold that are approaching that of biologics, meaning at least with our technology now, we don't believe that the cost of AAV is an issue anymore. I mentioned at the beginning that we have a collaboration with Vertex Pharmaceuticals. It's for two diseases, Duchenne muscular dystrophy and myotonic dystrophy. They had listed out for us predefined criteria to win additional success milestones. I'm very pleased to say that last year we gave them the capsid, meeting more than 17 different equations that they had given us to meet, and won the maximum success milestone, credentialing both our platform and our method, but also getting us the ability to go after helping these patients as well. So we are eager to see them progress these capsids into the program for Duchenne muscular dystrophy and myotonic dystrophy. Up until now, the only studies that they have done in DMD and DM1 have been not with our capsid, but with AAV9. Thank you for your time. I'm happy to take questions. I also hope that we are caught up back on time for the next presenter.